Krogan and many people from a consortium that there is to try to understand what host factors are required for viral replication for different viruses, including SARS-CoV-2. They are located at the UCSF. And what my colleague Nevan Krogan did early on, as soon as it was known the sequence of the genome of SARS-CoV-2, is identify all the proteins that are encoded by the virus, which is a total of 29 different proteins. And then he expressed these proteins individually in human cells, and then determined by finding the host proteins that bind to these 29 viral proteins, what host proteins are interacting with these viral proteins. This is what is called the virus-host interactome. And he identified a total of 332 protein-protein interactions. So what we postulate is that if a viral protein is interacting with a host protein, it has a good possibility that it does so trying to use this host factor for its own benefit, for viral replication. So we think that these 332 protein-protein interactions is enriched on host factors that are required for viral replication. And now among these host factors is the amine drug that is known, that is being used for another indication that inhibits one of these host factors. And with that, there is 69 of these different factors that they are druggable, meaning that they have some drugs, 1 or 2 or 3, that are known to inhibit this particular host factor. So we took all these host factors. I'm going to show you the one that is more promising in our hands, and how we led to a particular inhibitor that is now in phase 3 clinical trials. So this one of the factors is called EEF1A. It's a host factor. This involves normally in protein translation, in making our own proteins. It was found to interact with 2 of the proteins of the virus, with the SARS-CoV-2. And it was known to have some inhibitors that have been used or tried to be used for the treatment of some cancers, Ternatin-4 and Pritidepsin. So we took these inhibitors, Ternatin-4 and Pritidepsin, and we tested for inhibition of viral replication first in tissue culture, outside of the body, in a cell line. The cell lines that we used in the beginning were cell lines where the virus replicates very well. These are the Virus6 cells. It's an African green monkey kidney cell line where the virus replicates very well. And then we look, is this compound, Ternatin or Pritidepsin, these compounds, able to inhibit viral replication? And we tested multiple things, and not only these ones, but some inhibitors of some of the host factors. But this was some of the most potent drugs that we found that was able to inhibit viral replication. And then how we look at that is we look for ability of the virus to replicate in cells in the presence of different concentrations of these particular inhibitors, either Ternatin-4 or Pritidepsin. And by that, we can determine what is the concentration that is required from this factor to inhibit 90% of viral replication, which is what's called the IC90. And Ternatin-4 was able to inhibit viral replication with an IC90 of 10 nanomolar. Now, inhibitors usually are believed to be good when they're working at micromolar or submicromolar concentrations. So this is nanomolar concentrations, even 1,000 times less than when one tries to look for a good inhibitor. So that means that it's a very potent inhibitor. In addition, we look when it's able to have some cytotoxicity, and even though it looks like it's reducing cell viability in this broken cell, this doesn't, it does not kill the cells, and this is this reduction in percentage of live cells, 50% that we see here, is not due to killing of the cells, but it's because the compound prevents the dividing of the cells that happens in tissue culture, and therefore you finish with less cells in the presence of the compound than the ones that you finish if there is no compound. And Ternatin-4 is working very well, but even better, Pritidepsin, which is also targeting the same factor, also called the other name that has Apridine, and but it's the same compound, and the IC90 is in the nanomolar range, so even 10 times better than Ternatin-4. And the interesting thing is that Pritidepsin has been already approved for being used in in some countries, specifically in Australia, for actually the treatment of multiple myeloma. And it's been used combined with dexamethasone. So that means that it does not have a contraindication with dexamethasone, which is one of the inhibitors that is being used in immunotherapy for COVID-19. Well, so once that we find that it has activity in viral cells, then we tried multiple other cell lines, not anymore this African green monkey cells, but also we tried human cells, human respiratory epithelial cells, like these divided pneumocytes, but some of the cells like 293s. Basically, there are different bunch of human cells. And what we find is that aplidin or plitidepsin has a very good inhibition activity, again, at the nanomolar range for IC90s, which is even higher, meaning it requires less amount of the compound to inhibit 90% viral replication than remdesivir, which is at this time was the only inhibitor that was available for this virus. Is working very well also even in primary cells, primary human pneumocytes, which are the main target of viral replication in vivo, and is working even after 4 hours after infection of the cells, while remdesivir start not to be able to inhibit anymore 4 hours after infection of a cell. So it looks like, at least in these tissue culture assays, aplidin plitidepsin is more potent than remdesivir. Now, what we also try to do is really plitidepsin working by inhibiting this factor, the EEF1A. And for that, we are lucky in that it's known what area in E1F1A plitidepsin is binding to and inhibiting this factor. And not only that, because tumors, they also mutate very frequently, like viruses. There have been some tumors that they have been treated with this inhibitor of tumor growth, plitidepsin, and they have developed resistance by a single point mutation in EYF1A that prevents binding of plitidepsin to this factor. This is the amino acid 399, a change of an alanine to a valine. Now, in order to demonstrate that plitidepsin is actually working in inhibiting COVID-19 SARS-CoV-2 replication by binding to EYF1A, we make now, through molecular engineering, cells in which we change this single amino acid in EYF1A, and we make them now this factor resistant to binding to plitidepsin. And when we do so, now plitidepsin here in red is unable to inhibit any more viral replication as compared to the original cells. If we use these cells that have mutated EYF1A for looking to the inhibition of remdesivir, this doesn't matter for remdesivir. It does not bind to this factor, so it doesn't matter to remdesivir. But this demonstrates that plitidepsin is inhibiting viral replication of SARS-CoV-2 by binding specifically to EYF1A, and is not by binding to some other factor that drugs usually bind to more than one factor, and sometimes it's difficult to know if they have off-target effects. So in this case, it's not an off-target effect, it's by binding to EYF1A, how plitidepsin is inhibiting viral replication. Now, knowing that, then we started to go into in vivo. For in vivo, we use animal models of SARS-CoV-2 replication and disease. One of the ones that has been more useful for that has been mice that are transgenic for human AC2. The reason why we need to use transgenic mice is because mouse AC2 is not a good receptor for SARS-CoV-2. SARS-CoV-2 binds the spike to enter the cells to human AC2, and human AC2 is required for entry. Mouse AC2 is a little bit different from human AC2, and the spike of the virus is unable to use this receptor. But if you make mice to express human AC2, these are these transgenic mice, now the virus will replicate in mice and replicates actually to high titers in multiple organs. So we took these mice, these transgenic mice that were developed actually not for SARS-CoV-2, but for SARS-CoV, the first SARS, because it used the same receptor, so they can be used also for SARS-CoV-2. And then we infected with SARS-CoV-2, and then we looked to viral hepatitis at day 3. It's a very fast-growing virus in these animals. And we took some and leave it untreated, or treated with plitidepsin at a 0.3 mg per kg, 3 doses, or with remdesivir at 50 mg per kg, 3 doses, so a dose that is more than 10 times higher than plitidepsin. And when we do that, we get a comparable inhibition of viral replication, more than 2 logs inhibition of viral replication in the lungs, and also we don't get any more histopathology in the lungs. So we take these lungs, we look whether they look healthy. We have a score to show how sick these lungs are. And in the presence of plitidepsin, then we have almost no pathology of the virus causing in the lungs of this animal, indicating that it's working very well to prevent disease in mice. And with this data, PharmaMar, which is the company that actually developed plitidepsin as an inhibitor of myeloma, they have started clinical trials. This was the results from a phase 1, so a very small clinical trial where they looked mainly to toxicity that was found not to be toxic in the concentrations that were used. We used a concentration of plitidepsin that was based on the concentration that we knew that it was working in mice, and it has a similar behavior in humans with respect to concentration achieved in the lungs after taking plitidepsin. And there was only 50 patients per group, and not placebo, just 1.5, 2, and 2.5 mg per day for 3 days of these people. So again, no side effects. And that was at patients that they were hospitalized at the moment of hospitalization. And what we found is that at the higher dose, you can see that there is less days that the patients stay in the hospital as compared to the lower doses. And there is also a more quickly removal of symptoms from the people that from this very small trial. So there is a tendency here to a beneficial effect of treatment with plitidepsin. That's not completely proven because it's a small trial, there is no placebo, but that indicates no severe events and a tendency with a higher concentration to get the benefit. And with this now, there is a phase 3 trial going on, with now there is more participants, it's 109, and in multiple hospitals, where now there is also a placebo arm, so people are just treated with the standard of care, whatever is available in these hospitals. Many of the standard of care treatments is with dexamethasone for people that are being hospitalized, and with different amounts of plitidepsin in order to find out whether actually these 2 dose levels of plitidepsin versus control is reducing the number of days in the hospital of people with severe COVID-19. And we hopefully expect this trial to be finished by December 2022. So if that works well, then we'll have one more drug that could be used for the treatment of COVID-19 that could be added to the already existing drugs. And perhaps this will be beneficial because of you never know how many drugs you need for this virus and whether viruses are going to acquire resistance or not to the drugs that are already used.